everyone, in this video we will be discussing about the Arabinose operon. So whenever we are discussing about the operon system, we know that it is present in case of a prokaryotes, and in case of a prokaryotes, for the synthesis of the structural gene it needs a operon system, and in the operon system one promoter can control different type of genes. They are known as the structural genes. So here we will be discussing about the Arabinose operon. So before that we have to know what is this Arabinose. This Arabinose which is present, it is a 5 carbon containing compound and it is a carbon compound, so it will be utilized by the bacteria for its food source. So when this Arabinose will be utilized by the bacteria, whether in presence of glucose or absence of glucose, this Arabinose, which is a 5 carbon containing compound, it will be utilized by the bacteria in absence of glucose. When there is a lack of glucose, this Arabinose will be utilized, and it will be not be utilized in the Arabinose form. It needs to be converted into some other compounds for the utilization. So for converting the Arabinose into some other compound, it needs some of the structural genes. Here we will discuss about the structural genes which are responsible for converting the Arabinose, which is a substrate, into its product form. So the structural genes which are responsible, they are the ribulose kinase, isomerase, as well as the epimerase. So let's discuss about this operon system of Arabinose. So in case of a Arabinose, there are three things. They are the regulatory gene, controlling site, and the structural genes. The structural genes which are present, they are known as ARA-B, ARA-A, and ARA-T. And for this the promoter is PRA-BAD. So for synthesis of any of the genes, we need a promoter region, and one operator region is also present. In the promoter region, what the RNA will do is it will come and bind in the promoter region, and it will help in the synthesis of the structural genes. By the process of transcription, it will be converted into mRNA, and by the process of translation, it will be converted into the protein. So for the structural genes, this is the promoter region. So it is known as PARA-BAD. This BAD is Arabinose B, Arabinose A, and Arabinose D. So the Arabinose B which is present, it is responsible for the synthesis of ribulose kinase. Arabinose A is responsible for isomerase. Arabinose T is responsible for epimerase, and this is the promoter region for ARA-BAD. So the another thing which we have is the regulatory gene. This regulatory gene is the ARA-C, which is working as a positive regulator as well as the negative regulator. And when the concentration of ARA-C is high, then it will regulate itself. Now comes the controlling site. Which is the controlling site? Here we are having the controlling site ARA-O2, ARA-O1, and the ARA-L1. These three are the controlling site. That means the regulatory gene which will produce the protein, it will come and bind in this three regions. They are the ARA-O2, ARA-O1, and ARA-L1. These are the three sites, and this is the promoter region. This region is the promoter region for this regulatory gene, and this is the operator region for this regulatory gene. This is the operator region 2, and this is the operator region 1. And the one thing we are having, that is the cap. We will later discuss about what is the function of this cap. Now we are clear that it is having three type of genes. They are the regulatory gene, structural genes, and the controlling site is present. In the controlling site, the regulatory gene will come and bind in different region in different situation. So here we can see that what is happening when there is a presence of Arabinose and in absence of Arabinose. In presence of Arabinose, here there is a ARA-C protein. Here we are having two Arabinose protein. This is the Arabinose protein, and this is ARA-C, and they are in a complex form. When the Arabinose is present, it will help in the synthesis of the structural genes. The structural genes which are present, in presence of Arabinose, they will be synthesized, and in absence of Arabinose, they will be not synthesized. Why so? because if there is no structural genes, then the arabinose cannot be broken down. So whenever there is a arabinose, then only we need some of the structural genes, because it has to convert it into the usable form. Suppose we are having here a arabinose, this is a arabinose, and for breaking down or it needs to be converted into utilizable form. This is not a utilizable form, this is a Arabinose. This is not in the utilizable form, so it needs to be converted into utilizable form of Arabinose. During this situation, we need the structural genes. If this Arabinose is not present, the 5-carbon compound Arabinose is not present, we do not require the structural genes. So the synthesis of the structural gene is inhibited when the arabinose is absent, and when the arabinose is present, the structural genes is activated. So here we will discuss about 3 things. What happens in case if the arabinose is present? What happens when the arabinose is present? What happens when the arabinose is absent? And what happens when the arabinose is in high concentration? So before discussion, we have to see the structure of this arabinose. This arabinose, not arabinose, it is a Arac. We will discuss about the structure of the Arac. It is having a N-terminal region as well as a C-terminal region. We can see here 2 things. This Ara which is present, it is in the dimer form, and it is having one C-terminal region, and one N-terminal region. The C-terminal region which is present, it is responsible for DNA binding. So we are having a DNA binding domain. This is the C-terminal of Arac protein, and there is another thing, that is the N-terminal. Here we are having N-terminal region, and this is a place where the arabinose can come and bind, and these two are linking here because they will form a dimer. Whenever it is in the dimer form, it will work properly. So this is the structure of the Arac protein. The N-terminal portion which is present, it is responsible for dimerization. So now let's see how this arabinose which is present, it is converted into the usable form. This is a arabinose which is present. This is a intracellular arabinose which is present, which is a 5-carbon compound, and it is present in the bacteria. Now it needs to be converted into the utilizable form. So for the conversion, we require the structural genes or these proteins, that are the ribulose kinase, isomerase, and the epimerase. These are responsible for converting the intracellular L-arabinose into L-ribulose. For this conversion, we require Ra, which is a isomerase. This is a isomerase, and this is the interconvertible form, or it is a reversible form. And for this Ra, intracellular L-arabinose is a substrate and L-ribulose is the product. Now the L-ribulose which is present, it can be converted into L-ribulose 5-phosphate. Here a phosphate group is added. So for that we require the enzyme which is known as L-ribulose, which is synthesized by Arabi. And this L-ribulose 5-phosphate can be converted into D-gylulose 5-phosphate, which is a epimer. So for this we require L-ribulose 5-phosphate 4-epimerase, which is synthesized by RRD. It is again interconvertible or reversible form. So when this ribulose 5-phosphate and xylulose 5-phosphate is used, it is used in case of a pentose phosphate pathway. It is an intermediary product of pentose phosphate pathway, and it is used for the nucleotide synthesis. So in this way, this arabinose which is present can be utilized by the bacteria. So for that it requires the structural genes. Now let's see what will be how the condition in absence of arabinose. We saw that the regulatory gene which was present, that was the RRC, and it was in a dimer form. The RRC which was present, it was working as a negative regulator as well as the positive regulator, and it was a dimer. This is a situation when the arabinose is not present. When the arabinose is not present, it will come and bind in the controlling site. It is having the binding property. Now the controlling site are ARA-O2, which is the operator 2 for the arabinose, and the ARA-L1. In this 2 region, it will bind this RRC, the RRC which was present, it will come and bind into this region. So there will be a bending of the DNA. So when there is a bending of the DNA, the RNA polymerase which was responsible for the conversion of this RA gene into a protein, this RABAD, it was having the structural genes, that is the RAB, RA and RAD, which was responsible for producing the structural genes. It cannot be produced because for this bending property, when this RRC will come and bind in these 2 regions, there will be a bending, and for this bending properties, this RNA polymerase cannot bind in this promoter region of RAB, A and D. So there will be no conversion of this gene into the protein. Now let us see the other condition in presence of arabinose. Before that, we need to be very clear that this RRC which was present, it was coming and binding in this 2 regions. It is specific in absence of arabinose, that is the ARA-O2 and ARA-L1. This is the ARA-O2 and ARA-L1. In these 2 regions, the RRC will come and bind. This is the situation in presence of arabinose. When the arabinose is present, it will come and bind with the RRC. When it comes and bind in the RRC, there will be some of the structural changes of this protein. This RRC will have some of the changes in its structure. For that, it cannot bind with this ARA-O1 now. O1 now. It will come and bind in ARA-L1, that is ARA-L1 and ARA-L2. And during this case, when this arabinose is used, the concentration of glucose is low. We know that when the concentration of glucose is low, CAMP concentration is high. And there was a cap protein, cap region, which was present. And when the concentration of CAMP is high, it always binds with a cap region. And here there is a formation of a complex. And this will act as a activator for the synthesis of the structural genes. In case when there is presence of arabinose, the CAMP and cap complex and the RRC will work as a activator. It will work as an activator for the synthesis of the structural gene because it needs to convert the arabinose into the utilizable form. So now this RRC which was present, it is working as a positive regulator for the synthesis of the structural genes. Now what will be the condition when the concentration of RRC is more? We have discussed that it is a autoregulator. This RRC which was present, it is also working as a autoregulator. So when the concentration of RRC is more, it will come and bind in the ARA-O1 site. It was working as a dimer, and the dimer will come and bind in the ARA-O1 site, which is a operator for ARA. This polymerase will come and bind. This polymerase was supposed to bind in this region, and there will be transcription of this ARAC complex. But when this ARA-O1, in this ARA-O1, when the concentration is more, now let's discuss what will be the process when the concentration of ARAC is more. We have discussed that when the concentration of RRC is more, then it is working as a autoregulator. So the ARAC which was present, it was working as a dimer. the dimer which was present, it will come and bind in the ARAO1 site. So now the RNA polymerase which was supposed to come and bind in the promoter region and initiate the transcription process, now it cannot approach the ARAC promoter to work, because in the operator region it is coming and binding and it is having a negative regulator. When this ARAC which is present, when this ARAC is coming and binding in this region, it cannot help in the process of transcription of ARAC as well as the translation of this ARAC. So there will be no more synthesis of ARAC because the concentration of ARAC is already present in this cell. It is already high in this cell. So the main thing in case of this Arabinose operon system is when this regulator gene, which is the ARAC gene, is coming and binding in presence of this Arabinose, in absence of this Arabinose, or when the concentration of ARAC is more. This is all about this video. Thank you for watching this video till the end, and if you like this video, please do like, share and subscribe.